Thank you. All right, I like the enthusiasm. Nice. Because we're going to keep that momentum up, that tempo. There's a couple things I'm going to do today, and anybody that is late that's going to come tomorrow, I'm going to make it incumbent upon you guys to help bring them up to speed before I speak again, if that's possible. Here's why. Because today I'm going to present to you a paradigm that is not only 10 years in the making, not just in our lab, but globally 10 years in the making, and it addresses the over 200 years of undoing that we've been doing. And I want you guys to think about this paradigm, and I'm going to give it to you multiple times so that we can image it, we can have role-playing with it, we can see how it fits in your own clinical practice. By tomorrow, we'll be talking about protocols. So I will put this into play, and I will make this a lot more user-friendly than what I'm going to give to you today. However, what I need to make sure of today, because it is paramount, the way our healing dynamic is changing and the way it has changed over the last 200 years, and really in the last less than 50, if we don't do the things that are happening right now that I'm going to talk about today, not just with brain, not just with gut, but the bigger picture, the actual process will leave us behind. So we have an opportunity today. We have an opportunity to take everything we know and turn it on its head. So, without further ado, I want to tell you all that you are slowly losing your mind. That's not a euphemism for going crazy or stress or COVIDitis or whatever. We have a series of events that have been happening over the last 200 years where we are depleting, we are creating extinction events in your microbiota. How do we know this? We know this because of the last over decade of genomics-based research, which, by the way, we now do in our lab. We can go into the Amerindians in Ecuador or the Himalayas or Papua New Guinea, and you can look at these really isolated cultures. And they have more than 4 times the diversity of bacteria that industrialized nations have. And now we are starting to look at each one of those bacteria as individuals, which you'll see over the next 2 days, you'll see some of our individual bacteria. We're starting to learn what they do and what kind of benefits they can contribute to a healthy, productive, thriving organism on this planet. Now, conversely to that, what happens when you deplete or create an extinction event, whether it be a mammal in the ocean, whether it be a bacterium in your gut? What happens to the vacuum left, the hole left in that? It gets filled, doesn't it? What we're seeing in industrial populations is we are all losing critical organisms and we're replacing them with antibacterial resistant, pathogenic, inflammatory, and highly proliferative, meaning that they can grow rapidly in this new environment, and they change the way you crave, they change the way you feel, they change the way your happiness works. And guess what? Mothers are passing these organisms down to their babies. And one of the largest studies ever done on infants demonstrated that industrialized countries, about 30% of the women in industrialized countries are missing Bifidobacterium infantis. That is one of the early colonizers of babies. So when they look at babies, and it's a combination of things that happens. 1, way overuse of antibiotics. 2, cesarean sections, C-sections, cesarean surgery. And 3, there's nothing in our environment, there's nothing in our products, there's nothing in our daily lives that will reintroduce these extinct organisms. So there has been movements, by the way. There's been a number of companies, including ours, that have gone out and isolated these Bifidobacterium infantis and are going to be offering them in forthcoming products. Furthermore, breast milk is the milk that feeds the very specific dynamic of early colonizers of babies. So prebiotics is another theme for today. Prebiotics change the way bacteria behave, change the way they can grow, and they can exclude some of these pathogenic or non-commensal organisms. They can essentially squeeze them out. And we'll get into a little bit of that later. We'll unpack these packages as we go. However, what I want to really push home today before I get into the brain, now that I've told you you're losing your mind because you're losing bacteria, which control your mind and your happiness and your choices and your cravings, and that's been demonstrated over and over in animal models. There are organisms we can give to an animal and force it, that organism sends messages, neurotransmitters to the brain, and now that animal wants to eat nothing but one kind of food. Good example of that I'm going to show you in a minute is toxoplasmosis. Have you guys heard of that? That's an example of how a bacterium or a parasite or a fungus can hijack the neurological centers of our brain. But how I want to turn this and flip this upside down for you, because by raise of hands, most of you probably have heard the term microbiome, right? To how many of you is it still a little bit of an abstract idea? Is it more than just probiotics? Is it more than just going to the grocery store and getting a probiotic? Or, I can't remember the names, the psyllium husk, or, the flavor of the day prebiotic or fiber. It's so much more than that. And where have we learned all this? In the last decade. The last decade has transformed how we think about it. And I'll give you so many examples. You're probably ad nauseum, so you can call uncle when I show you the clinical study after clinical study. And I'm not even going to bore you with all the studies we've done in our lab, although they're very amazing. I'll give you a little snippet tomorrow. Because we can isolate and study these organisms individually and then put them back into a collective, or what's called a consortia of beneficial bacteria. And they come from many places, like the soil. They have their own purposes. I won't get into that today. All of that being said, I know that's a lot to dump on you all at once. I want to paint a picture of how I want you to think about my talk the rest of these 2 days. Have you guys ever heard of thinking of the microbiome as a beautiful, lush rainforest or a beautiful garden, and you have to nourish both the terrain, which would be the soil, and the microbiome would be these lovely, healthy, vibrant plants? Is that analogy somewhat approachable and you can understand it? Here's what I want to flop on its head. I want you to think of the bacteria and the soil together. And now I want you to think of the human as the flora and fauna in the garden. Is that clear? So instead of the soil being the terrain and the microbiome being the plants, I'm now pushing the soil and the microbiome together, or the prebiotic, probiotic world together. And now this amazing flora and fauna is the human. Because every choice we make now regarding our nutrient intake, our dietary intake, our exercise, I want you to put it in the perspective of a microbiota. If you're going to muscle test, I want you to ask questions about the microbiota before you start asking questions about neurotransmitters. If you're going to use functional testing, I want you to start thinking about, do a gut questionnaire. We will offer microbiome testing soon, so that will get you covered down the road in about 6 months. But I want you to put everything in the context of the microbiota and then ask the physiological questions. Ask the nutrient questions. Ask the questions about minerals, vitamins, organic acids, small molecules, mold, any of those questions, parasites. Ask the question in the context of the microbiota. That's what I want to send you home with today and tomorrow. Because I can prove to you that the thread that connects your nutritional status, your neurotransmitter status, your hormonal status, your ability to fight off parasites, your ability to generate and sustain new mitochondria, the thread all leads back to the microbiota. Without question. That data is it's incontrovertible. How much of the control of the microbiota, that's forthcoming. The more we learn, the more we get our tools, the more we have our pre's and our pros. But everything I've said is incontrovertible. Everything is connected through the microbiota. So no matter how you practice, in your practice, the first question I want you to ask is, I wonder how my patient's microbiota is doing. And then I can assess nutrition, emotion, parasites, heavy metals, detox. Some of the most important detoxing phenomena happen on the surface of the bacterium in your intestines. A lot of what we know about polyphenolics and flavonoids and alkaloids and all these things that we've grown to love as herbologists, they all get transformed by the microbiota before your body sees them at all. And if you have the wrong microbiota, they get transformed into toxins. Or they get converted. Heavy metals can be converted into methylated heavy metals by microbiota. Otherwise, they were harmless prior to that. And so on. Pesticides, same thing. Phthalates, PCBs, and so on. I had a model here, and it's collapsing because of the light, I'm thinking, because it was doing better earlier. And what I'm trying to indicate here, and I'll draw it for you, because one of the important parts of this model that I'm trying to impress upon you by making everything go through the lens of the microbiota is because everything goes through the microbiota literally. It's not a figurative. It's not simply my idea of how to envision the world. This is the magnification of the inside of your colon, small intestine, doesn't really matter. The skin has a very similar representation. The epithelial tissues in your nose, in your lungs, all throughout the respiratory, all throughout the urogenital tract for women. You have this lining that is covered in mucus. So for example, this is the villi, right? There is a mucus layer that is produced by your goblet cells. And most of us can appreciate this, whether or not you get a runny nose or just in general. Every lining that is exposed to the world has a mucus lining to it. Furthermore, it is rich with a microbiota. And it's up to us to decide whether that microbiota is a healthy one or an unhealthy one. Which means when you have this really thick layer, and remember, these are one cell thick, and we'll get into that in a minute. There's only one cell between most of your epithelial tissue and everything inside your body. The nervous system, the blood, the immune, and so on. But this thick mucus is filled with, guess what? Microorganisms that you don't go into our body without moving through a field of microorganisms. This is the microbial aura as such. Nothing gets into you without going through that wall. Now, if that wall's broken, we have leaky gut, we have problematic organisms, it goes right in, doesn't it? There's tests for that. We've seen certain organisms open right up tight junctions, which opens up skin tight junctions, which opens up lung tight junctions, which opens up brain tight junctions. The phenomenon is not a unique phenomenon anymore, and we're seeing it more and more in our population because the microbiota is no longer healthy. 300 billion tons of antibiotics have been distributed in the last 10 years, I believe. Right now, on average, an American citizen or European gets 40 rounds of antibiotics before age of 30. And just recently, a study was released that if I give you one round of an antibiotic, your microbiota, at least both intestinal and on the skin, those are the only two sites measured, will not recover for 3 to 4 years fully. One round of antibiotic, and that's JAMA. That's not our data. That was done by a large consortium of microbiologists in the microbiome world. Now, they were shocked because they have been arguing, and we've heard this before, that your microbiota recovers quickly, right? How many times have we heard that? Oh, stress, or I can take an antibiotic and my microbiota goes right back to normal. Absolutely false. And they stopped the study at 4 years, so they don't even know when it actually did recover. The younger that happens, the more damaging it becomes. Although, it doesn't mean that at 3 years of age or 4 years of age, you can't do anything about your microbiome. That's absolutely false. Again, that's another fallacy that's been created. There's so much control we have over it. Now that I've got you set up to putting everything through the lens of a microbiota, I'm going to be going through some of my slides rather quickly because I don't think I anticipated being so long-winded on the intro. All right? But you know, you guys are tough. You guys are smart. You guys have been down this road. And you've certainly heard me sing this tune a number of times. Perhaps too many times, but with this new lens, by the way, you're going to be having to listen to me on little videos, on our website, on social media, because this is the wave of the future to deal with healthcare. And what I'm going to try to remember to tell you is you don't assimilate methylation products as well if you don't have the right microbiota. You don't assimilate iron. You don't assimilate magnesium. That's the lens I want you to think about the new way of doing things, whether you're a muscle tester, whether you use BioMeridian, whether you're a functional medicine. That's the new lens, and I can prove most of that to you, and I have research ongoing that's going to prove the rest. So please hit me with questions and say, hey, I don't believe you. I'm all about that. All about it. And we have new scientists in the lab. We now have a couple of PhDs. We got a new geneticist that is running up the microbiology and the DNA testing. So we're really excited to bring you guys some cool stuff. So I've pretty much killed this for you at the moment. Some stats. We love stats. It gives you the impetus. It tells you this is a crisis. We know it's a crisis. I just want to remind you guys all, this is a growing crisis, and the latest pandemic did not help the crisis, by the way. The stats have gotten worse. Oops, where's my backwards? I'm not going to bury these, but psychological diseases, neurological diseases, I might use that term interchangeably because they can be related to each other. Although one deals with more of the psychology basis of symptomology, whereas the other one is neurological. They're interchangeable in many cases, at least in my language today, and we can tease those out if you'd like. 5 million Americans suffer from Alzheimer's. That's a growing trend. 400,000 from MS. And the reason I'm telling you these are neurodegenerative diseases that I'm going to link to the microbiome today. 30,000 from Huntington's. If left unchecked, and these are really casual statistics, these are not aggressive. We're going to miss the mark on these by a ways, but 12 million Americans will suffer from a neurodegenerative disease in the next bit. We're already seeing it. We're seeing it reach into younger and younger children. You guys agree? Are you guys awake? All right. So, the second brain. Many of us, this story goes back hundreds of years. I'm going to hit a few people that made some amazing predictions, by the way. There's some scientists from the 1700s, 1800s, 1900s that saw a relationship between neurological, gut, and psychological outcomes. They saw it happen. There were some really brilliant people, and they made a quote, they got it published, and no one cared after that. Furthermore, that was pre-antibiotic era. So furthermore, not only did we not listen to some of these amazing people, we trashed it afterwards. You know, we went the opposite direction. But the second brain is an amazing central nervous system. I don't want to spend a lot of time on this because I know most of you are familiar with the anatomy. However, it's a giant nervous system, and it innervates every nook and cranny of every organ in your GI, as well as lungs, as well as kidney. It's pretty amazing how it works. And it's not only designed to essentially bring the nervous system to your organs, it happens to be designed around the microbiota. It speaks directly to the microbiota. Now, I need you to believe me on that, because the science that has shown that would really bore you guys, and I would start down that path pretty easy, okay? I don't want to do that to you, but it's pretty amazing, and we can test that hypothesis regularly, because certain bugs, you throw them into a mouse, you make it produce a metabolite, you put it in mRNA, and you can actually turn on genes and muscles and so on in the brain. Not muscles, but you turn on all these neurons in the brain with one organism. Now imagine the 1,000 species you have, which is shrinking, but anywhere from 300 to 1,000 species, all talking to your brain via the enteric nervous system. What's another way we could test it, or we have tested it? What's one way to test whether or not the enteric nervous system has a direct line to the brain? What's the large nerve that carries the enteric nervous system to the brain, center of the brain? The vagus nerve. Cut it, see what happens. No, don't do that, don't cut it. But they do that a lot in animals, and they've shown that absolutely works. So it's connected by the vagal nerve, I already told you that, neurotransmitters, and so on. It's 200 million in size. It's really quite beautiful and elegant. This is what it looks like. Get my laser out. Excuse me. This is what it looks like, and the reason I want to point this out, and the reason I have this up here, is because there's one cell away from 200 million nerves. There's only one cell that separates all the bacteria in your gut to all of the systems in your body. I want to point out in here that every system, you guys, my little experiment didn't work so well. It's kind of collapsing. But this is the one cell thickness. This is your entire bloodstream. This is the enteric nervous system. This is your central nervous system. This is, again, the blood. This is all of the immune cells, goblet cells, enteric cells, enterocytes. Your bacteria is in constant communication with every system of your body right here. And that's repeated in the lungs, that's repeated in the mouth, and so on. It literally has it has the VIP pass to everything you do. We'll get more into that in a second. Okay, you guys probably seen this a number of times. I want to highlight this because by tomorrow, we're going to be talking about specific species and strains of organisms. And the world of the microbiome has been able to find a lot of new ones. And as I mentioned, we're doing studies across the globe on native people that are isolated versus people that are right in the middle of New York City. In New York City, we've found anthrax, plague, and so on, all in the subways. Now, yeah, it's kind of fun for those of you that don't mind them. But again, you can see there's a lot of, these are just a short list of the potential 1,000. And the more healthy and diverse you are, the more you have of these basically family, genus, species. We can get into that tomorrow if you'd like, because in the probiotic world, that gets really confusing. And maybe if I have time tomorrow, we can deconvolute some of that, because it is important. Not everybody is doing the work, but there are a lot of probiotic companies taking advantage of it. So we do want to keep in mind that there are people that see this science and it's amazing and it's coming out every day by really credible people showing amazing results. And there are companies going, hey, that's cool, let's just buy up probiotics. Here's why I bring that up. We are buying clinical probiotics that have clinical research behind them, and we are having our own clinical probiotics. And together we're joining them with prebiotics. However, I've had to spend quite a bit of time on the phone with the only 6 about probiotic companies that create clinical probiotics. And I've had to tell them what our mission is, because there are other companies buying up the entire stocks of probiotics right now as we speak. Companies with a lot of money are just buying up the stores and putting it on Amazon Prime and Amazon whatever. So I've had to be working with the scientists in these companies, explaining to them what we're doing, so they don't sell their entire 100,000 pounds to Joe well, I almost said a bad word, Joe, John, Sam, whatever. So it's been a struggle, and we're now creating relationships with these companies as well as growing our own. So this has been quite a really fun journey over the last decade. And we'll get more into that tomorrow. But you can see there's a whole bunch going on here. That's not something new. I've shown you that before. The discovery of this was in the 1700s. So Marie Bichat discovered this second brain. How brilliant is that? If she could have maybe, raised a flag back then to all of our ancestors, we'd be like super brilliant right now. But we've had to relearn everything that she discovered back then. The galt, right? We all know the galt is part of the gut. These are all things that I just want to remind you of before we get deep into the bacteria of it all. But the autonomic nervous system, the central nervous system, the enteric nervous system, they all collide here, which means the various bacteria have the ability to communicate with central nervous system, autonomic, or enteric. That's why we talk diversity. If you end up monoculturing or having dysbiosis or SIBO or all the other terms that we talk about, it's no wonder therapies stop working with these people or don't work well or have a short-term benefit. And the reason I'm really pushing that lens bit through the microbiome is we have personally experienced people that were on, let's say, on the spectrum, and we started them off years ago on the cellular formulas, and we saw amazing improvements for a while. And then that plateaued and started to decline. Back then, I wasn't looking through the lens of the microbiome or trying to understand how methylation pathways are significantly changed by the microbiome and not by just providing specific nutrients. Does that make sense? You can provide all the methylcobalamin, methylfolates, and so on. If you don't have a healthy microbiome, you're getting a temporary fix at best. That's the lens. What does the system look like? It's pretty fun in terms of we can now call it the microbiome gut-brain axis. So it's no longer a gut-brain axis, microbiome gut-brain axis. And some of the early studies, and when I say early, we're talking 5 years ago. This is a new science. This is brand new stuff. We learned really quickly that it controlled the HPA axis. The microbiome can control the HPA axis. And it starts in really young people. That control begins right after birth. And I won't get into all the details, but if you guys just Google that, you're going to have 1,000 papers come up. You're going to be bored, and then we can just call me and we'll go through them. It'll be fun. How does it do it? It runs in a number of ways. It has afferent sensory as well as efferent. They can go both ways. The gut communicates with, or the bugs communicate with the brain, and vice versa. So the brain can dictate what bacteria it wants in your gut, and vice versa. And the brain gets really frustrated when it wants certain bacteria in the gut and they're not available. The brain starts to activate in weird ways. So it's a bidirectional relationship that we've got to be very, very cognizant of. But it happens in a number of ways. There's cytokines, there's lipopolysaccharides, and what I can tell you now is it can control cortisol, hormones. That we now have bacteria that we know stimulate the hormones. We can have bacteria that can stimulate estrogen, progesterone, testosterone, cortisol, CRP, and so on. Furthermore, I'll show you in a second that there are now bacteria that also produce neurotransmitters. So all your neurotransmitters you're getting and signaling do not have to come from your own cellular tissue. There's a number of organisms that have learned to evolve with us that produce things like epinephrine, dopamine, serotonin. In fact, serotonin, there's more serotonin made by the bacterial GI than anything else in your body. That is the highest concentration. And it's driven by the bacteria in the gut. So how fascinating is that, you guys? I don't want to lose you. And I'm not going fast enough. All right, Nobel laureate Menshikov, another brilliant man who identified in 1907 that colonic flora were associated with behavioral issues. And so he made this quote, if we could change the colonic flora, we can change the outcome. what if we could do that and we could, have really wise elderly people because their colons were cool. That's just a fun thing. And a quote, this is my quote actually, so it's not great. I got to work on my quotes, but the microbiome has, and it should be I don't know how the there came from. See, look, I already misspelled. The microbiome has an immense influence over health. Central to this powerful and elegant relationship is how it affects all aspects of our senses, including happiness. It does have an influence on your happiness, among other things. Now, another great slide for you guys to really get stressed out about. This shows you the simplicity of the layers, but the complexity of what can happen to the brain. When this is broken, we have potential disease states, all of the ones I've listed, and we'll show them in a second. All the causal factors, here's a short list of them. Toxins, drugs, stress, exposure to environment, and it could even be pathogens. Sometimes a parasite can trigger this if your microbiome is not ready for that parasite. They can trigger a cascade of events. Here it is again in a different way. Remember I told you that the microbiome can control hormones? These are 3 hormones that it controls quite readily. CRH, ACTH, and cortisol. You can take in an animal and swap out the microbiome and change the concentrations of those 3 hormones very quickly. You take a germ-free mice, you throw in a new microbiome, you can ramp those up. You give him antibiotics, throw in a new microbiome, you ramp them down. The microbiome is controlling so many of our hormones. What else does it do? It produces GABA, norepinephrine, dopamine, serotonin, all through tryptophan metabolism for the most part. So what are the fun stuff that it does? Well, I mentioned earlier today that we're worried about it with ASD, Crohn's, ASD with Crohn's is the gut side, but MS, Parkinson's, ADHD, and so on. We're worried about these really serious diseases. What else fun about it? What else is fun about it? Right here. Cravings, happiness, relationships, intimacy, peacefulness, mindfulness, and wisdom. You can actually alter many of these states through a healthier and unhealthy microbiota. And there's many articles on this. Most of it's done in animals, but also many in humans. In fact, some of the probiotic companies have just finished depression studies on a double-blind, placebo-controlled, and they've shown that they can alter the depressive states of young people, 20s and 30s, through just altering their microbiota through a very few number of organisms. Now imagine if we take a lot more and have a lot greater diversity, what we can do to these types of things. Now cravings is easy. That's done over and over again. We see that in the animal world all the time where a microbiota changes a craving. We certainly see it in what we call metabolic syndrome. When you take the microbiota of overweight people with type 2 diabetes, metabolic syndrome, and you lay that microbiota out compared to a healthy microbiota or someone that does not have metabolic syndrome or type 2 diabetes, etc. When you look at those two, compare and contrast the bugs, and then throw those organisms into an animal, and actually one time it was in a young lady in Britain, got a fecal microbial transplant from someone that had metabolic syndrome, and she sued the doctor because she gained 50 pounds in like 6 weeks because of the microbiota that she took on. So cravings clearly, and now we're seeing it in the gene expression. So when we look at the gene expression of the human compared to the gene expression of the microbiota, we're seeing a lot of craving genes turned on by the microbiota. So the microbiota is sending messages into your body saying, eat more sugar, eat more chocolate. You probably saw the CNN article about the chocolate cravings where they showed that certain people that have chocolate cravings have certain organisms in their gut that leads them to want more chocolate. And you're going to see a number of these fun little things come out because they send these neurotransmitters in. Some of these organisms, imagine if every time you eat chocolate, your organism produces dopamine. So what are you going to do next time you eat chocolate? You get a dopamine hit. Ooh, let's try that again. Dopamine hit. Ooh, The organisms can do that. In fact, they'll do that even more than heroin in animal models, right? They'll get this dopamine hit from a food source. Moving on. Okay, pathogen model. I wanted to touch on this. I mentioned earlier because the toxoplasmosis. But we know, for example, Toxoplasmosis gondii, it hijacks, when it infects a mouse, it creates a bunch of neurotransmitters, hijacks the mouse, makes the mouse really bold and cocky and testosterone-filled, so the mouse marches out and picks a fight with the nearest cat and gets eaten. Why? Because toxoplasmosis wants to be inside the cat. Doesn't want to be in the mouse. So it hijacks the mouse, gets eaten. Now it can reproduce in the cat and make more of itself. Isn't that cool? Now, men with toxoplasmosis are less conscientious and experience personality changes. This is documented. If you know someone in your life that has toxoplasmosis and they're male, please deal with that parasite because they're aggressive, they're non-empathetic, they can be actually, their lives can be turned upside down and they don't even know why. And that's one example. I can give you probably 20 of those. It's happening all around us. So pathogens are a problem. What's the best way to fight pathogens? The absolute most powerful way to fight off pathogens. Say it louder. Thank you. Who said that? The best defense you have against anything, toxins, parasites, other pathogens, is what nature intended. And that is a diverse microbiome which produces antimicrobials, antifungals, antiparasitics. It creates all of these amazing things we're talking about. We've trashed that microbiome over the last 50 to 100 years, which means you can't do the fighting without it, which is why we're experiencing a lot of things now, and that's going to continue, which is why I'm being way more animated today than I'm normally animated. I'm normally a science guy, but this is the beginning of something that is going to change our world, without question. If we don't start putting the microbiome first, I don't care how many organic acids you take, or how many methyl bombs, or how much of our product even. I love our cellulars, but they will not do their job if the rest of that system is broken. And that's a system that was designed with you and for your ancestors and for the planet 1 million years ago. It was designed into you. It's designed into every living being. Okay. All right. Guess what else? How many people in here have had babies? I have not. I have children, but I have not had a baby. What is one of the gross things that happens when women give birth? They poop. How long have we been trying to make sure the baby avoids that or wipe off that meconium? That's been an ongoing thing, right? There are some European hospitals that changed that about 5 years ago after learning that every mammal, they've caught whales, when the baby comes out, it reaches around and it eats poop. Dolphins, turtle, every organism gets a little dose of poop as soon as it comes out. Why do you think that is? Because of the microbiome. It's got to be set up. Whales, dolphins don't have a chance, or deer, or they come out. You've seen elk born up in Yellowstone. They have like 2 seconds to survive. They're out, they eat a little poop, they run because there's wolves everywhere and so on. This is not something that we need to take lightly anymore. This is a serious thing, even though it is fun for me and I love it. And I wish the lab people could be here because we'd all be talking shop up here and you guys. But anyway, the lab people love it too. And we've been working with a few people that are called, mainstream medicine, because we have to work with universities to get some of the research done. We have literally converted a few MDs with this subject. Like entered into this going, what, you guys, this microbiome started 10 years ago and it's a pipe dream. You guys remember those kind of things 10 years ago? It's not going to amount to anything. What is it? Now, guess what? They're writing grants. They're trying to get funding. They're trying to do what we're doing because they see what it's doing to their patients, what it's doing to the globe in general. The globe in general. China is adopting it. Why is China adopting it? They're putting billions into it right now. Which is one of the reasons I'm so animated and I want you guys to get on this because if we don't do this through our industry, we are going to be swallowed up by the pharma industry. There are now 4 drugs on the market that are microbiome based. 4 drugs. If they keep getting away with that, they could potentially come to me and say, Shayne, you've got these 4 bugs. We think they should be drugs. You can no longer give them out. If we don't get ahead of this curve, and the 4 bugs by themselves are great, but they're nothing without the consortia and the prebiotics. So they've still missed the mark. They're getting great results with people, but they've still missed the mark. They're not offering a prebiotic. They're not offering a consortia. They're still narrowly thinking about the problem of a diverse microbiome. You guys are not, are you? At least not after today. My voice is going to be ringing in your head for days. So anyway, it's really fun. Pathogen model, we did that. Beyond antibiotics. I've kind of hit this a little bit, and I mentioned that they did a study recently, which I thought was quite shocking. So what they did is they looked at a bunch of drugs and why we were killing our microbiome so quickly, and why it seems to be accelerating in concert with all of these autoimmune issues growing at an exponential rate. Things are accelerating, and they're not linear. They're starting to accelerate, and we don't know why. So we start looking into what are people taking? What is so much of industrialized people taking? Well, they looked at 835 drugs, 156 were antibiotics, and others were painkillers, blood pressure pills, etc. And a quarter of them were accidental bactericides. SSRIs. They kill your microbiome. Blood pressure pills, kidney pills, all these things that have been warfarin and others, they're bactericidal to your microbiome. So we're not only killing it with the antibiotics, but everything going in there. And then you add to that plastics. And I'll never understand the bottled water phenomenon, I'm sorry. The plastic is dangerous unless you do your due diligence. And like I use glass, I use stainless steel, so on, but plastic also is an antibacterial. It leaches, phthalates, PCBs, so on, they leach. There's so many things we're onslaughted by. We're so much of an onslaught to our microbiome. And the microbiome is a canary in the coal mine. And even further than that, you've got something called phage in your body. They're more sensitive to environmental changes than the bacteria. We offer 4 phage. I don't have time to talk about them today. I want you guys to look into them because they're brilliant. And I wish we had 30. I wish we had 40. But these organisms are even weaker and they keep your bacteria healthy. So when you lose them, you're also losing a segment of your healthy population. So it's the microbial diversity hypothesis. Instead of thinking about antibiotic resistance or the hygienic principles, think diversity. Spin it on its head. Stop thinking of the negative side, which is this. It exists. It's real. It should shock you. Start thinking diversity when you're with your patients. Think diversity in the way they eat, diversity in the way they take supplements, diversity in their microbiome. That's going to generate a new way of thinking. We want something called the diversity hypothesis, not the antibiotic resistance hypothesis or the sterile hypothesis. Does that make sense? Think diversity. They're all associated, and I am really not going fast enough for you guys. A long history of GI and psychiatric neurological associations. This is what led neurologists and psychologists and psychiatrists down the path, which, by the way, they are now teaching in school, the idea of a microbiota. Because almost in every pathogenesis of a neurological or a psychosomatic or a psychological issue, there's a GI issue. Very rarely do you get somebody that has autism or ADHD or so on that doesn't have a GI issue or a dietary issue and so on. It's very rare, and if they do, it means they haven't looked closely enough and then they figure it out. So they're intimately associated, which started leading certain microbiologists to say, wait a second, there's not only association, there's a causation. There's more than just a correlation. We're now looking at causation. This slide should have been way earlier in my talk because we've already talked neurotransmitters and hormones and all the things that microbiota does to contribute to this. When you have dysbiosis, IBS, IBD, all these things, guys, you are likely going to, if not already, have some sort of a neurological issue. It's just a matter of time. Even as things as simple as diarrhea. Persistent diarrhea in third world countries end up over here. It's very sad in many places. All right, what are the exacerbating deals? Leaky gut, inflammatory disease, toxin assaults. These add to the problem. Pathogens, autoimmune issues, and so on. There is a segment of this that is our own genetics. I'm not going to get into that today. Some of our other speakers, I hope, will do a little more on that. Let's hit a couple of diseases quickly, because I've really laid the ground. You know mostly about the microbiota and why it's important. But there's been a number of studies done in 3 areas. There's more, but the 3 I'm going to get to you today. Parkinson's is one of them. So it's a complex breakdown of the ecosystem. You lose diversity in Parkinson's patients. What's next is there's the microbiome theory was first presented. We've pursued that. There's an influence over neurotransmitters. Clearly, there's an influence over neurotransmitters now. Furthermore, there's inflammatory microbes. When you look at a Parkinson's patient, compare it to a healthy patient, they have a different microbiota. They have a dysbiotic one. They've allowed the wrong bugs to get in there for a number of ways. You'd have to look at their life to see how it happened, but the microbiota is no longer healthy. They have a lot of reactive oxygen species microbes, and guess what? Microbes produce a protein that triggers alpha-synuclein plaquing. So Parkinson's is alpha-synuclein plaquing, and guess where it starts? I think I have a thing for this. So there's environmental toxins. We talked about that. This article was really good if you want to read it. It goes into it was done by the gastrointestinal liver physiology group. It's well done on a Parkinson's study, and they were looking at the GI motor, all the things that the microbiota contributes in a Parkinson's patient, and how when that goes wrong, a lot of things start going wrong in those patients. Now there's hope, right? If it's so intimately tied to microbiota, can we do something about it? Abso-freaking-lutely. Okay. Alpha-synuclein accumulates and initiates in the gut and propagates via the vagus nerve. So the plaque protein that ends up in the brain actually starts plaquing in the gut, and then it attaches to the vagal enteric nervous system, and they can actually watch it migrate up the vagal nerve over time. There is a small subset of Parkinson's patients that they have there's a genetic predisposition, they're slightly altered, but the vast majority, when they clip the vagal nerve, they stop the progression. Some humans have gotten their vagal nerve clipped that are starting to progress down the Parkinson's pathway. Not everyone statistically, but there's a lot of them that benefit by doing that because it begins in the gut. And it turns out, after all this research, some of the microorganisms that we found in Parkinson's people, it's been discovered that you can see not only the disease progression and then, of course, the limited motor problems as that synuclein gets up into the brain, the impact, the actual microbes start to create this plaquing in the gut. We want to get rid of those bugs, don't we? We want to replace those bugs with healthy bugs. They've been identified now. So down the road, when we do microbiome testing, we're going to have on our panel some of those bugs that we know create alpha-synuclein plaquing. Now, that's a powerful tool, and I don't want to make too many crazy promises because there's always a complexity for each and every one of these. But look where we are now, when we had no idea 5 years ago where in the crap these diseases were coming from, how to deal with them, what to do about them. And by the way, everything we've done in the last 2 decades, none of these diseases have gotten less frequent in the population, have they? They're all escalating. They're all increasing in an exponential way. So all the stuff I've been doing and everybody else, we are not getting ahead of this. We have to get ahead of it. We haven't gotten ahead of it. I can tell you right now that's not happening. And it's starting to show up in third world countries now, where they were one time isolated. And that poor Amera Indian tribe I told you about, the French went in and gave them vaccines, by the way, after they were discovered. They took their microbiome. It was brilliant. They published it. It was amazing. And they thought, well, we better vaccinate them all. I just learned that the other day. This is very sad. Sorry. Total off key. So the microbiome relationships. Enterobacteriaceae, that is positively correlated with PD. We don't like Enterobacteriaceae. We want less of them. B. wadsworthia, a keystone species for sulfite production, is also correlated positively with PD, meaning we don't want that in our gut. So we can look for these down the road, right? We don't want those in there. These show up. They do us dirty. Let's switch gears real quick. The same scenario, we don't have to go through it in detail, but the same scenario exists for depression. A very similar etiology, similar breakdown, similar concept as the PD. Depression can be exacerbated, if not triggered, by gut microbiome. It alters, again, neurotransmitters. It alters the way the gut motility happens, which can lead to hormonal fluctuations. Cortisol can increase, and so on. You can kind of see this in your mind, right? I don't need to go through all the mechanisms because those we know, now we know that the microbiota influences them. So again, what I want to point out that now in the context of depression, it's linked to a healthy or non-healthy microbiota. What else can we look at? Autism spectrum and the microbiota. Again, a number of publications have really been digging into this, showing some really amazing results. But the other piece on this that you don't have in depression is there are some genetic things you can also look at in some of these people. It's not strictly microbiota. It's not that simple. I wish it were. But the microbiota, genetics, environmental factors can all contribute. But now we know there is at least a microbiota component, and they're getting some improvements. Some of the most recent clinical studies have shown that at least a big chunk of patients improved when they got them on a regimen that improved their microbiota. Not everyone. Some are resistant to improvement, which means we got to try something else. Could be nutrient deficiency, could be genetics, whatever. But it worked for a big subset of many clinical studies. So that's pretty exciting. I find it really exciting. We can break it down to specific bugs between the gut and the brain. I mentioned that the bad ones. There's good ones. Phacobacterium prasinutzi, Mycobacterium vaccae, a couple of lactobacilli, reuteri, Johnsoni. We can break it down now and start looking at the bugs that are beneficial, whether or not they produce serotonin or dopamine or acetylcholine. Some of the soil-based organisms that we're now using produce neurotransmitters. How cool is that? That's an association, that's a relationship that we developed a long time ago, and we never thought of them as even commensal. We thought of them as transient. You eat something out of the ground, it moves through your body, it's got dirt on it, you take a carrot, it moves through. We think, oh, it's gonna leave. They don't leave as quickly as we thought they did. So there's a lot of good stuff there you won't get from me today. That's gonna have to be a later talk. Sorry. Probiotics altering the paradigm? Yes, they are. We're seeing a lot of amazing publications. Here's a short list, and I encourage you to maybe just read the abstracts. If you get down into the dirty of it, it can be pretty, here's the genetics, here's how many genes we pulled out, and whatever. It can really get thick on the reading, but it's really fun. We can change the intestinal barrier. We can change all the things I mentioned today. This is probably a snapshot of 1/1,000,000th of what's out there. So it's crazy cool. We're working through mechanisms. We only have a couple in the lab we can do because of the tools, but we're starting to see how it's working, how it's modulating the immune system, how it's modulating or producing. We can see the genes in the bugs that produce dopamine or produce hormone mimics. Many of the hormones that they produce are actually hormone mimics. So bacteria figured out how to make a mimic of our hormone and then send that into your body. It's pretty cool. Mechanistically, it's fun. I know this is really heavy science for you, but I am going to bring it to a conclusion. So in diabetic rats, again, these are some really cool studies I want you to look at. Take this home. You can read this later. Another one out of the indigenous microbial colonization can affect development of brain plasticity. That's the HPA axis. You can retard plasticity if you don't give people early in life the right microbiota. I wish I could go back 25 years to my children and know what I know. Because I would do things differently 100%. 100%. Therefore, I'm telling you, and I'm telling everybody I talk to, which means I want you to tell everybody you talk to, these stories. This is transformative for young people and old people. Old people, what's the one thing that happens to the older generation? Their microbiota gets simple again. You start in life with a simple microbiota. Okay, this is simple, this is complex, this is simple. You start life with a simple microbiota, which is good because you're infants. You're only eating one thing, breast milk, hopefully. It gets really complex, but then in the geriatric years, it gets simple again. Why? That's when the disease states accelerate, by the way. They don't have to be simple. When you look at people in these really remote regions of India or Italy, their microbiome is the same as a 20-year-old. And they live like a 50-year-old, and they're 100. We're doing this to our elderly, guys. We put them on simple diets. We think they need mono diets, potatoes, and we're shifting the thinking. They need to be eating every bit as diverse, crazy food as the 20-year-old. They need to be experimenting, eating hot peppers, throwing up, whatever. They need to be doing that for a diversity purpose because their microbiota is leading to a lot of the problems that we see in the elderly population as well. I keep getting off on topics, you guys. You got to stop me. So we can access psychotropic-like properties. We call these psychobiotics, neurobiotics. It's pretty exciting. Love doing it. You're going to see more of that tomorrow. Here's one of the ways we do it. We look at all the different pathways up and down. Neurotransmitters, I already told you this, but here's some hard data for you. It tells you which bugs, by the way. So if you're curious, there's lactobacillus and bifidobacterium that can do GABA. Escherichia, Bacillus, and Saccharomyces can produce norepinephrine, and so on. These are pretty cool bugs that we're now playing with. Luminal administration, let's see. These are, again, just data. I want to thank you guys because I don't even know my time. Am I over by that far? Oh, I got time. I forgot the clock was down there, you guys. I looked at my slides, but I didn't look at the clock. This is fantastic. Yeah. I got really excited. Super excited. All right. What I want to get into, because if you remember on these slides, a lot of this data is just that. It's data. It's teaching us that the theory that we generated 10 years ago, 11 years ago, the hypotheses that came out of the microbiome studies. And by the way, those were on the heels of the human genetic studies. You guys remember that? When we became genomic, and we could start finding all the genes in a human, what are they doing, all the genes in an elephant and a mammoth in the ice melt of Siberia, we got really excited about genomics. The microbiota kind of was born out of this, and it stuck. A lot of the other science is really fun to know and kind of to trace, but you can't do everything through human genomics. And I love human genomics, by the way. But how many genes do we have in our body? Around 23 to 25,000. Now, when we first sequenced 3 billion base pairs in every cell of your body, and we only found 25,000 genes. Now, there's a lot of other stuff in there, by the way. Tons of it. And that's called dark matter, and that's coming out. Some of it is actually ancient viruses. Some of it is what we call transposons and things. I won't bore you with that. But 23 to 25,000 genes, every scientist took a step back and said, that's not possible. It's wrong. Let's do it again. First time they did it, cost them $1 billion to sequence a first human. Everybody thought it was wrong because there is no way 25,000 genes can make you who you are. Corn has more than you. C. elegans worms have more than you. I mean, almost everything on the planet has more than us, except for bacteria. So the Microbiome Project filled the gap. How did the immune system work? We didn't know. I was in a group of immunologists in graduate school, and they had literally hit a wall. We don't know anything more about the immune system because they were studying all the immune cells. And they've studied those for 20 years. But they couldn't figure out how the immune cells were learning or how they were getting their information. Guess what filled that gap? The organisms in and on our body. As soon as they discovered that, then millions and hundreds of millions went into, let's figure out the microbiome. Now, the first shock was everybody's is slightly different. Unlike the genetics, where you're all 99.9% identical, the microbiome was very different. Now, there were some pieces, like familial. If I were to test my microbiome and all my people in my family, we'd share a lot together. We'd have a lot of associated organisms because we live together. And there'd be some that my mom gave me and some that my dad gave me that I've kept. Because I don't do antibiotics. I can throw that off with the antibiotics. But the real cool thing about this regarding the genetics of us as humans, guess how many genes exist in the microbiome in your body right now? How many genetic pieces of information can you tap into as a human being for everything? For the air you're breathing right now, and it's got mold in it, I can tell you right now. For the stress you're feeling because I'm talking at you. Or the joy you're feeling because you now love the microbiome as much as I do. All of these genes that are being turned on and off in your body, there are 3 million to 9 million microbial genes that make the functionality, survival of you possible on this planet. You remove those genes, you don't live. Your 23,000, 20,000 will not sustain you on this planet. You won't make it a day. The 3 million genes you take advantage of metabolize every plant you eat. They make enzymes that you've never even thought of. They make all the mimics, all the protective bacteriocins and antibiotics. Your gut is a cesspool of antibiotic production by your own organisms. And by the way, antibiotic resistance is not a scary thing. You know why? Even the Amerindians or the Hadza, they all carry bacteria with antibiotic resistance. Why? Because antibiotics are part of nature. They're produced by fungus, bacillus species. They're produced by penicillium and so on. No matter where you are on this planet, you're getting exposed to them, which means the bacteria in your body has to deal with it. Naturally. They're natural. You don't want to supplement them. Antibiotics are not a supplement for any of us. However, these 3 million genes transform everything that you're exposed to, everything that you deal with on this planet. If you guys all came to Utah. Maybe some of you are native Utah, but how many of you notice the air's a little drier? You got a little less oxygen? Guess who's helping you adapt to that right now with your mitochondria? Your bacteria are helping you to adapt to that. Not your own cells. They don't have nearly the ability to adjust so rapidly. Athletes, really cool Harvard study, and Harvard actually is going to patent this, by the way, which kind of bums me out. But they discovered in endurance athletes an organism, allotypes. And in endurance athletes, they test for it at the beginning of the race. They take a stool sample. Then they collect stool samples throughout, so these athletes are pooping in their pants as they're going. But They collect these stool samples. Allotypes goes from being about let's say, you've got trillions in here. Let's say allotypes is hiding in a corner and there's about 50,000 cells in there of this bacterium. By the end of the endurance race, there's 1 trillion allotypes in that gut of that endurance runner or bicyclist. Did you know why? It metabolizes lactic acid on the go. So the best athletes have more of that organism that goes way up during the athletic event. They're finding that also in people that are developing big muscles. They're finding a very different group of organisms that harvest every stinking amino acid those guys and gals eat. They've trained or changed their microbiome to absorb every amino acid and throw it into the muscle. But they're huge, right? You've seen these people. And they're strong, and it's amazing to look at. But I like too many sports that that movement would restrict me. I don't want to be restricted. I like to wakeboard and that kind of stuff, so I don't want to do that. But the microbiome is central to that physiological phenomenon. And I could keep going. The 3 million genes that you now have in your body are either doing you a benefit or a disservice. That's the lens of changing the human organism through this next 10, 15, 20 years. And I would encourage you, any chance you get, to talk to a practitioner that uses antibiotics or these other compounds, just encourage them. Plead. Be very political if you need to. There's a small city in Sweden city in Sweden that they've gone through and they've gone through a whole big marketing campaign saying, only use them when you've diagnosed it with a bacterial infection that is life-threatening. So they have cut the use of antibiotics by over 40%, and they're seeing dramatic improvements in every stratus of their population. They're not finding it in the water. They're not finding it on the animals. They've already put a moratorium on the use of it in chicken and cattle. You guys know that's where we use a lot of it, right? I know that now I've kind of gone a little political on you, but every time you drink water, even if it's a plastic bottle of water or whatever, even through some RO systems, you could potentially be exposing yourself to antibiotics. And your patient certainly could be. And every time that runs through you, it's disrupting the microbiota every single time. Now tomorrow, keep in mind, and you need to take a grain of salt, tomorrow I'm going to talk to you about our approach to creating diversity and feeding that diversity through our own research. And as we do that, I want you guys to frame that in a protocoling mindset. I'm going to tell you by the end of tomorrow, and I want to repeat it again, so I don't mind saying it today. If I want to start off a patient, and I know there's a microbiome issue, I'm going to run them through a whole series of the pre and probiotic protocol. And I'm going to rotate it. And I'm going to coordinate it with possibly their testing when we have it, stool testing, whether you're muscle testing and so on, and I have some ideas there. You rotate. You think about their questionnaires. You could do an intake questionnaire, and I think we have one on the website. You also look at circadian rhythms. If they're in an area like Utah or anything above a certain latitude, then you're going to take certain probiotics and prebiotics and use those in the wintertime, and then use other ones in the spring and other ones in the fall. You're going to start to rotate this protocol and create a much more dynamic change. Because in the past, each and every one of us, our microorganisms went through a rise and a fall, and they still do. And for some people, that can be monthly. In women, that rise and fall of microbiota happens every single month. When a woman gets pregnant, it's really awesome. First trimester, she creates a unique microbiome. By the third trimester, that microbiome shifts to a very caloric, intense microbiome. It's trying to save every calorie mom eats. That's why in the third trimester it's tricky. It's why you have sometimes gestational diabetes or a lot of weight gain and so on, because the microbiome can shift And if you're not careful by being more diverse in your thinking, that shift can be unhealthy for pregnant women in this current situation. Does that make sense? We're seeing it. I mean, there's a lot of really cool microbiome studies on pregnant women. It's really amazing. But again, these are all things I want you guys to teach and train and think about with your patients. There isn't a time and a place that you shouldn't consider the microbiota, whether it be on the skin, whether it be in the lungs, whether it be in the eyeballs, whether it be in the nose, whether it be in the gut, whether it be in the urogenital tract. Down the road, we'll talk about fertility. Microbiome dictates fertility. If you have an incompatible microbiome, you have a much higher probability of miscarriages and non-fertility. There's all of these little pieces that will go, you'll take the journey over the next year and deal with. So it's pretty exciting, and before I let you go, I want to answer questions. I want to engage all of you in some ideas. I had a pretty long talk. I bounced around a little bit. Tomorrow will be more protocoling centric. I hope at least, by the raise of hands, does everybody understand my one goal today was to frame the microbiota in the lens of where we start, not where we get sidetracked. We start there. Does that make sense? And I can demonstrate over time that all of our herbs, all of our nutrients need to be in that framework. Now, that makes it easy to protocol. We will protocol through a pre-probiotic scenario, and then you can add in really fun and unique nutrient or herbalomic or other compounds depending on your genetics or on the symptoms you're treating and so on. If you have the framework with microbiota, you can start to plug in a protocol fashion all these other things, and you're going to get better results. Much better results. Your consistency is going to go up. You're going to have fewer people going, I don't notice anything. I'm taking this really amazing multi and I'm not feeling any better, or, there's a reason for that. So we have to get down that journey. We have to begin with avoiding the bad things, right? Getting our microbiome back in shape. And the truth of it is, by tomorrow in the product side, we have a lot of amazing tools, but we are not even beginning to scratch the surface. So there will be times that you'll run into cases where you're going to be winging it. You're going to be taking tools that we've given you, other tools, you're going to be combining things, because we certainly don't have the answer for everything, and it's going to take another 20 years to do that. But we will. We will get there. And we will transform health care as we know it. Do you guys have any other questions? Yes. Oh, microphone.
Testing. Whoa. It's alive. That's really live. Thank you. And this might be for tomorrow, and so if you don't want to answer this today, that's fine. But for a patient who's had an event like a stroke or a heart attack, and now obviously they have neurological concerns with that, and then we know that their biome is off, and we know that their bad bugs go to sources or sites of injury.
Right.
I'm interested in if there is protocol specific for them, or if their protocol is just the same, or,
There's subtle differences. Now, I have a protocol tomorrow that I call the cognitive protocol. And the first part of it is universally applicable. You start with the neuro, immune, and then we rotate into the other 5 or 6, sortings that we have. However, you can target things like neurobiome or neurovision. But one caveat I want to tell you guys all today, and I'll reiterate this tomorrow. Anytime we have an injury or something like autism spectrum disorder where we know the gut is a wreck, we have to start with microdoses. You take a scoop of that, and you guys are going to get them tomorrow, a scoop of the prebiotic for neuroimmune, the Terrabiome, which is the terrain building one. You take a scoop of that and give it to someone that has a wrecked microbiota, you might give them all kinds of cramping, gassy, frustration, and you don't want to do that because it throws them off base. I've been dosing everybody at work on these products recently. I got a couple of fart complaints. I didn't get any, and this is a mostly healthy population though, so I didn't get any major bloating or major cramping or anything. Not everybody's done it yet, and I'm going to continue to move it through people. But other people I've microdosed. Instead of taking a full scoop, I start with 1/4 of a scoop. And in a case like this, I would do that. I would start with the prebiotic and the probiotic at the smallest possible dose you could do, and then graduate it up. Because it is going to change the overall behavior of the microbiota. Even so, the probiotic is going to only deliver 20, 30 organisms at a time. The prebiotic is going to stimulate the 300 species you've got in there in different ways. And as it changes the behavior through the oligosaccharides and so on, and we'll get a little more of that tomorrow, you're going to have some organisms struggling. Those are the ones you want gone. They don't metabolize these things. They love sugar. They love starch. As you start shifting that, they're going to start getting angry. And they're going to be sending messages to the brain. This is what happens. You start getting these messages from these microorganisms saying, knock it off. Go back to the donut. If you don't, I'm going to make you sick. And they will. They will cause a little bit of havoc. They might cause a little diarrhea. They might try to bloom at the last minute, right? SIBO, they might try to bloom in the gut and create a whole bunch of trouble before they die off. They'll make a last stand. They'll do the Alamo. They are there to survive. And they've taken up in you to do such. We need to shift it to the ones that we've evolved and are happy with, not the ones that have figured out how to take advantage of us and our poor diet or whatever. So yeah, you start small and you'd incrementally increase that, and you'll start seeing a benefit of the diversity kick in. And then you add in some of the nutrient supplements, and they should all be really well assimilated at that point. Yeah, but we can cover some of that tomorrow too. Somebody else had their hand up a second. Come on, you guys, you got to bring it on.
Hold on.
Okay, so I have 2 questions. The first one is, the organisms that are being patented, are these naturally occurring or have they been genetically modified in some way?
Yeah, that's a great question. And that is a fight that even we are putting our hat in the ring, because what I didn't mention, you guys, what's exciting is we now have the 3rd patent on our pre and probiotics. So how did we get patents on pre and probiotics? Because we're patenting the unique organisms along with the unique food that feeds them. Does that make sense? We're not patenting the bug. You own your bug. This bug was actually pulled out of a number of people in Utah that live on farms and things. So a lot of our bugs are locally sourced from the local poop. Some children, by the way, we got some really cool ones. But they're their bugs. I can't patent that bug. So that is a huge fight right now. What they might be doing, and this is why you'd have to dig deeper into this if you come across someone prescribing some of these drugs, is they might be genetically modifying them. Then you can patent it. So what came right out of the human, you can't. But what we did is we took all these, we've got 20 unique organisms now out of humans, and we've got over 70 different plant materials. And we're not talking just macronutrients. We're talking polyphenolics, flavonoids, alkaloids, all these really cool things that we found in the lab. This stuff made this stuff grow. So when you guys eat, you're not eating for just you. We figured that out. We tried for years to get these human organisms to grow outside the body. And the brilliant scientist by the name of Michael, full credit, I'll never understand how he did it, he started taking random plants and growing them in the lab with these bugs. Glutathione's one of them. One of our organisms needs glutathione. How cool is that? One of our other organisms needs another organism, needs a buddy. One of our organisms has to have a bifidobacterium in culture or he won't grow, or she, whatever. The process to get to where we got literally took 11 years. And I'm really amazed by it, and I wish I could. If you guys ever come to the plant, we'll show you a lot of that, but you can't see it. And I didn't want to present that data tomorrow because it would take all day. But what we're seeing is, in order for us to get these probiotics to work, we're essentially looking at all the things that we know are amazing for what we've always thought were amazing for our own cells, our own mitochondria, our own Golgi apparatus, our own membranes, all of that. Those same compounds allowed us to grow the probiotics that are good for us. Is that not cool? Methylcobalamin. What else did we figure out? The N-acetylcysteine. One of our bugs loves N-acetylcysteine. There's just so many. Tryptophan. I mean, there's just so many of these really cool things. And by the way, the plant polyphenolics, those things end up becoming, soil when plants break down. The polyphenolics, the isoflavones, and all those things. You guys have heard of these words like genistein, rutin, all these really cool terms you hear about. The benefit they give us, turns out, is nowhere even near us. When we watch what happens to those compounds, they're all fully metabolized by the bacteria and then sent into the bloodstream looking completely differently than they started. Resveratrol, have you guys heard of that? That's completely transformed by healthy bacteria into these compounds that are really good for us. If you don't get that transformation, you guys had any people say, well, I'm taking resveratrol and I'm taking like 500 milligrams and it doesn't do anything. They don't have the right bugs. It's not affecting their sirtuin genes at that point. There's some really cool stuff coming down the pipe. And I'm really happy to give it to you guys, even though it can be heavy. It can be a little bit, minutia. But if you walk away with a little bit of this, you're changing your lens to a brand new lens. And I'm telling you, the more you look, what's the cognitive biases, the heuristics, the phenomenon of what you put out in the world you're going to receive? Now that I've told you guys all about the microbiome, you're going to see it everywhere. You're going to look at people and go, man, I can tell what microbiome you've got. If you're a muscle tester, and I have a really cool thing to talk about this with someone later, you need to ask the questions about microbiome first. Microbiomes will prevent parasites, or they'll live in harmony with parasites. We have lost that ability here in a sterilized environment. If I were to transplant any of us over to India, rural, how long would we survive? Not very long without puking our guts out, would we? But many of them do beautifully with what we consider pathogens. How many of you guys have heard of the Hadza or the Amerindians, and we look at their microbiome and think, they're amazing. I can tell you guys right now, if I transplanted their poop into most of us, we would have a really bad day. Because we don't live the way they live. We don't live in the environment that requires some of those organisms to do the kind of metabolic processes they do. We have transformed enough that we need to find some balancing in the middle. None of you guys are going to become hunter-gatherers in the next little while, are you? None of you are going to kill your animal and eat it raw right there in the field, like they do. And they eat the intestines, and they eat the feces, and they do all that. That's not likely. I don't see too many of us doing that tomorrow. So the microbiota reflects that behavior. But does that mean ours is good? No, ours is complete crap. Theirs is beautiful. Theirs is keeping them alive and healthy and thriving until they get a hold of antibiotics. But we can find a new version of our microbiota. We know what it should look like for the most part. There's going to be some more information coming out. But we've got a good start, you guys. This started 10 years ago. My first microbiota talk was over 10 years ago. And even then, I was hesitant to make any really strong claims. I am no longer hesitant. The data has been flying in over the last decade. Flying in. I mean, just go search microbiota, whatever, microbiota in brain, microbiota in poop, or whatever. You're going to get thousands of articles. Not everything is really amazing, well done, but a lot of it really is. There's some associates that we have in the field, and these guys are leaders in the field. Rob Knight is one of them. Right? He has traveled the world and studied every kind of feces on the planet. He is a feces guru. And by the way, when he was down in, whether it was Africa or down in Papua New Guinea, his microbiota changed. While he lived and ate what they ate, he looked like them. But guess how long it took to change when he got done, when he came back to the States? back to California. That's even better. It reverted back to a not as good a microbiota, although he's really healthy and he's conscientious of his microbiota, obviously. But it became more industrialized in about 60 days after he got back. Is that a bad thing? No, because he doesn't live out there. However, what he did identify is he's missing critical keystone organisms. So he's replacing organisms in his own body, which is what we're doing. This takes me all the way back to the beginning. Guess what? We're losing our mind. Why? Because we have extinction events in our microbiota. They are missing. They're missing in us. They're missing in our children. We would have to go probably to great-grandpa to get them back. And I can't do that. They are no longer here. However, we can start replacing them as we learn how to do that and keep them fed. Is anybody here? I worry about animals' extinctions. Do you guys not? Polar bears are on the brink. The blue whale was, and we're losing what, a bird species a month or more. Does anybody know some of this? We are losing extinction. We're creating extinction events in animals on this planet. You're also creating extinction events on the animals in your gut. We don't even know how many. The estimates has been as high as maybe 10 a week. Where it used to have 1,000 species, the Hadza tribe had like 1,600, almost 4 times what a normal. Anyone in this room probably has about 300 species. Ideally, we'd like 1,000. So we could be missing, any one of us could be missing something like 200-300 species that our great-grandparents had. We got to get them back. There's just no question about it. We're going to get them back. And I still didn't go over. It's a wrap.